Hello dear learners, welcome to APCC LMS portal. I am K. Sai Sudha, LMS presenter working as lecturer in biotechnology at DK Government College for Women in Nellore. In this video, we are going to discuss on a topic, enzymes involved in DNA replication. Before going into the topic, let us see the learning objectives. By the end of this video, the student will be able to understand the complex enzymes involved in DNA replication. You would be able to explain the importance of DNA polymerase. You would be able to understand the mechanism of DNA ligation. You will be able to discuss the helicase and topoisomerase enzyme activities, and you would understand the importance of SSB proteins, and you would understand the synthesis of primer. As we have seen earlier, DNA replication is a fundamental process for transferring genetic information from one generation to another. There are several enzymes and proteins that are involved in DNA replication. Few of the important enzymes or proteins are DNA polymerase, primase, DNA ligase, helicase, SSB proteins, and topoisomerases. Here, we are looking into the enzymes or proteins that are involved in prokaryotic DNA replication. Let's see the DNA polymerase. DNA polymerase is an enzyme that synthesizes new DNA on a DNA template. Hence, it is also called as DNA-dependent DNA polymerase. It requires a DNA template for the synthesis of new DNA. Both prokaryotes and eukaryotes have different DNA polymerases. In prokaryotes, there are three important DNA polymerases: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. DNA polymerase 1 is the first discovered DNA polymerase. It was discovered by Arthur Korenberg in 1960, for which he has got Nobel Prize. Hence, it is also called as Korenberg enzyme. DNA polymerase 1 is a single polypeptide chain with 1,03,000 Dalton's molecular weight. It is encoded by poly A gene in E. coli. DNA polymerase 1 catalyzes the addition of deoxynucleoside triphosphate to a growing chain. So, one DNTP is added and one PPI, that is pyrophosphate, is released. This DMP is added to the growing polypeptide chain. The formation of phosphodiester bond occurs by a nucleophilic attack of 3' hydroxyl group of a growing polypeptide. DNA polymerase 1 is the first discovered DNA polymerase. It was discovered by Arthur Korenberg in 1960, for which he has got a Nobel Prize. Hence, it is also called as Korenberg enzyme. It contains a single polypeptide chain with 1,03,000 Dalton's molecular weight. It is encoded by poly A gene in E. coli. DNA polymerase 1 catalyzes the addition of deoxynucleoside triphosphate to a growing DNA chain. It adds DNTP, and in this reaction, pyrophosphate is released. The reaction occurs by the nucleophilic attack of 3' hydroxyl group of a growing DNA strand on the alpha phosphate of the incoming deoxynucleoside triphosphate. This forms the phosphodiester bond. This reaction is catalyzed by DNA polymerase 1. There are certain requirements for DNA polymerase enzyme for its activity, such as template, primer, and the substrates. The template is a very important aspect because the DNA polymerase can only synthesize DNA based on the sequence of the template. The DNA polymerase enzyme requires a template, and it is template-guided DNA polymerization. The new nucleotides are added complementary to the template strand. And this DNA polymerase also requires a primer. Primer is a short segment of RNA or a DNA, which is elongated by this DNA polymerase enzyme. The requirement of primer is very essential because DNA polymerase cannot initiate a DNA synthesis by itself. Hence, it requires a pre-existing strand that is synthesized by primase enzyme. Primase enzyme synthesizes a short segment of a primer, which is RNA rather than DNA. And DNA polymerase also requires the substrates deoxynucleotides. They are DATP, DGTP, DCTP, and DTTP. The DNA polymerase 1 has three important activities. The first one is 5' to 3' polymerization activity, which is the main activity of the DNA polymerase, that is for the synthesis of DNA. Second one is 3' to 5' exonuclease activity, where this DNA polymerase can remove the nucleotides from the 3' end. This activity is very important for detecting and removing the mismatched nucleotides, and this activity is called as proofreading activity. The third important activity is 5' to 3' exonuclease activity. This activity is important for the removal of RNA primers. So this last activity is unique for DNA polymerase 1. So DNA polymerase 1 has three important activities. These three activities are present in three domains of the DNA polymerase. For example, say this. This is DNA polymerase 1's structure with three important activities. While a mild proteolytic digestion of DNA polymerase may produce two fragments. One is a larger fragment and the other one is a smaller fragment. So this larger fragment will retain two important functions, that is 5' to 3' polymerization and 3' to 5' proofreading activity. These two activities are present in the larger fragment. This larger fragment can be used in our DNA technology, because it has two important activities. This experiment was done by Klenow, and hence it is called as Klenow fragment. The smaller fragment will retain the 5' to 3' exonuclease activity, which is not useful. So the larger fragment is called as Klenow fragment, and it has wide range of applications in DNA technology. In spite of all the activities, there are certain drawbacks of DNA polymerase 1 to be the prime polymerizing enzyme. They are: polymerization rate of DNA polymerase 1 is very slow. It adds only 600 nucleotides per minute, which is very slow. It has a very low processivity, that is, it has only 3 to 200 nucleotides processivity. Processivity is nothing but it is the number of nucleotides added before the polymerization enzyme detaches from the template. So this DNA polymerase 1 can add only 3 to 200 nucleotides before it gets detached from the template. So it is a very low processing enzyme. And the most important drawback is the mutants of DNA polymerase 1 is viable. That means in spite of inactive enzyme of DNA polymerase 1, the cell is able to survive. That means some other enzyme is the principal enzyme which is involved in the synthesis of DNA. These drawbacks led to the search of other DNA polymerase enzymes. Later, due to these drawbacks, DNA polymerase 2 and DNA polymerase 3 were discovered. But what is the function of DNA polymerase 1 in the cell? The DNA polymerase 1 is involved in the removal of primers after DNA synthesis, and it plays a very important role in the DNA repair mechanism. Then comes the DNA polymerase 2. DNA polymerase II is a multimeric protein with a molecular weight of 88,000 Daltons. It has two activities, 5' to 3' polymerization and 3' to 5' exonuclease activity. So the polymerization and the proofreading activities are present in DNA polymerase II. The polymerization rate of DNA polymerase II is 40 nucleotides per second, which is better than DNA polymerase I, but it is not sufficient for the principal DNA polymerizing enzyme. Its processivity is also very low. Its processivity is 1500 nucleotides. This DNA polymerase II is also not the principal enzyme for DNA synthesis. This is involved in DNA repair. Then comes the DNA polymerase III. DNA polymerase III is the prime DNA synthesizing enzyme in prokaryotes. It is a much more complex enzyme than the other enzymes. It has 10 subunits and a total of 17 subunits per enzyme. The core enzyme of DNA polymerase III has 3 subunits, alpha, epsilon and theta. Alpha, epsilon and theta. So, alpha subunit has 5' to 3' polymerization activity for the DNA synthesis. Epsilon subunit has 3' to 5' exonuclease activity, that is for proofreading, and theta subunit has unknown activity. So, DNA polymerase III also has two activities, polymerization and proofreading activity. But the 5' to 3' exonuclease activity is only present in DNA polymerase I but not in DNA polymerase II or III. So the core enzyme along with beta sliding clamp and clamp loading complex is called as holoenzyme. So the DNA polymerase III holoenzyme has three parts, core enzyme, beta sliding clamp and clamp loading complex. So the holoenzyme of DNA polymerase III has three parts, core enzyme, beta sliding clamp and clamp loading complex. Core enzyme has three subunits, alpha, epsilon and theta. Alpha subunit is encoded by the gene POLC and it is involved in DNA synthesis with 5' to 3' polymerization activity. Its molecular weight is 1,29,900 Daltons. Epsilon subunit has a molecular weight of 27,500 Daltons. It is encoded by DNA Q gene and it has proofreading activity. Theta subunit has unknown activity. It is encoded by Hol E gene. Then comes the beta sliding clamp. Beta sliding clamp has a molecular weight of 40,600 Daltons. It is encoded by DNA N gene. And the major function of beta sliding clamp is to increase the processivity. The processivity of polymerization, DNA polymerase III is infinite. That means DNA polymerase III binds to the template and it will not detach from the template until the complete synthesis of DNA takes place. This infinite processivity of DNA polymerase III is due to beta sliding clamp. So there will be 4 beta sliding clamps which are attached to the holoenzyme of the DNA polymerase. 2 beta sliding clamps attached to each strand. Then comes the clamp loading complex. This clamp loading complex are involved in loading the beta sliding clamp onto the template. So the clamp loading complex has 6 types of subunits. They are gamma, delta, delta prime, psi, psi and tau. Gamma subunit has 47,500 Dalton's molecular weight. It is encoded by DNA X protein and it has clamp loading activity. The same is the case with delta and delta prime. They are also involved in clamp loading activity. Psi subunit is involved in the interaction with SSB proteins and psi subunit is involved in the interaction with psi and gamma. The tau subunit is very important protein or subunit where this is involved in dimerization of the core enzyme. That is, the two tau subunits are attached to two core enzymes and this tau subunit dimerizes these two core subunits, core enzymes, and it will not allow to detach themselves. So you can see the number of subunits here per holoenzyme. This is the molecular weight of those subunits, and these are the genes responsible for encoding these subunits, and these are the functions of the subunits. So a total of 17 subunits are present in a holoenzyme of DNA polymerase 3. So DNA polymerase 3 is the principal DNA synthesizing enzyme in E. coli or prokaryotes, and it has 17 subunits. Then comes the DNA ligase enzyme. DNA ligase enzyme catalyzes the formation of phosphodiester bond between 5' phosphate of one strand and 3' hydroxyl group of another strand. So this enzyme actually ligates two fragments of DNA which may be formed due to any endonuclease activity or any breakage in the DNA. So a small breakage in the DNA causes a small nick, and these nicks are sealed or ligated by DNA ligase enzyme. So this DNA ligase forms a phosphodiester bond between two chains of DNA, and it is an endergonic reaction. The DNA ligase enzyme is important because, for example, if this is a DNA strand, here the nick is formed due to the breakage of a phosphodiester bond. Since here in the 5' end only one phosphate is present, spontaneous formation of a phosphodiester bond cannot take place. Neither DNA polymerase enzyme can synthesize a phosphodiester bond because only one phosphate is present in the 5' end of a fragment. Hence, DNA ligase enzyme is very essential. Here, let us see the mechanism of DNA ligation. Let us consider this as a DNA ligase enzyme. This DNA ligase enzyme needs energy in the form of ATP. It requires ATP for its activity. So the first step of DNA ligation is the enzyme DNA ligase binds itself to the ATP. So ATP will be cleaved into AMP and pyrophosphate. Pyrophosphate is released. AMP will be bound to the DNA ligase enzyme to form adenosine DNA ligase complex. This is called as adenylation. So this AMP DNA ligase complex will go into the near to the neck, and then the enzyme will transfer its adenosine AMP to the 5' phosphate of one fragment. So what happens is the AMP will be transferred to this phosphate. Whenever the phosphate has an additional AMP, then the OH group which is present on the preceding strand will act on this phosphate nucleophilically and forms a phosphodiester bond. Here, the AMP is released out and the DNA ligase enzyme will be released out. The nick is sealed with a phosphodiester bond. This reaction is catalyzed by DNA ligase enzyme, and this DNA ligase enzyme is very important for binding the Okazaki fragments after their synthesis in vitro. And this DNA ligase has potential applications in RDNA technology. The DNA ligase enzyme requires its energy from NAD or ATP. In bacteria, NAD plus will act as an energy source, whereas in animal cells and viruses, ATP will act as an energy source. Then comes another enzyme, primase. Primase is a monomeric protein with a molecular weight of 60,000 Daltons. It synthesizes a short fragment of RNA primer at the 5' end of the newly synthesized DNA. Primase is a type of DNA-dependent RNA polymerase. So, DNA-dependent, that means it requires a DNA template, and RNA polymerase, it synthesizes a RNA. It is a type very similar to RNA polymerase enzyme, but it synthesizes only a short fragment of RNA. The primase activity is of utmost importance since the DNA polymerase cannot initiate DNA synthesis by itself. DNA polymerase can only elongate a pre-existing DNA strand but cannot synthesize or start the DNA synthesis. So, DNA primase enzyme is very, very important. Single primer is synthesized in leading strand, whereas multiple primers are synthesized in lagging strand. So, the two templates which are present, one is called as a leading strand and the other one is a lagging strand, because leading strand has a continuous DNA synthesis, whereas lagging strand has discontinuous DNA synthesis, and in lagging strand multiple primers are produced in front of each fragment. So, this is the activity of DNA primase enzyme. Then comes the helicase enzyme. Helicase enzyme is a hexameric protein with identical subunits, and it has molecular weight of 3 lakh Daltons. It is encoded by DNA B gene, and hence it is also called as DNA B protein. Helicase is involved in unwinding of double-stranded DNA to form single-stranded DNA. So, this is due to DNA polymerase cannot unwind the DNA itself. It requires helicase activity. It unwinds DNA by cleaving the hydrogen bonds between the two strands of the DNA, and it requires ATP for its activity. So, helicase enzyme will continue to break the two strands of the DNA, and by breaking the hydrogen bonds between them, so that single-stranded DNA is formed. This single-stranded DNA acts as a template for the synthesis of new DNA. Then the next one is single-strand binding proteins, SSB proteins. These are tetrameric proteins with identical subunits. They have a molecular weight of 75,600 Daltons. The major function of SSB proteins is to bind with single-stranded DNA, which is formed after the helicase activity. So, you can see here, this is the helicase enzyme. Helicase enzyme will break this hydrogen bonds and forms a single-stranded DNA. This is the single-stranded DNA. This is one DNA and this is another DNA. Single-stranded DNA has a natural property of pairing with each other. That is, it has complementarity, so it will instantly reanneal or rewind itself. So, to prevent the rewinding of the single-stranded DNA, SSB proteins comes and binds to this single-stranded DNA. They prevent this reannealing or rewinding of the single-stranded DNA. So these SSB proteins bind to the DNA until the DNA polymerase comes and synthesizes new daughter DNA. Whenever the DNA polymerase comes into the vicinity, the single-stranded binding proteins will be displaced from the single-stranded DNA. So this SSB proteins has a function of binding with the single-stranded DNA and preventing the re-annealing of this single-stranded DNA to form duplex DNA. Then the last enzyme is topoisomerase. Topoisomerase is a very important enzyme which helps in relieving the DNA from topological stress. This is considered as a reversible nuclease. It cleaves the DNA and it will join the DNA. This enzyme relaxes the supercoils formed in the DNA due to the activity of helicase enzyme. So whenever the helicase enzyme unwinds the DNA, there will be formation of supercoiling ahead of the replication fork or ahead of this helicase activity. So this supercoiling has to be relieved, because unless it is relieved, the DNA further cannot move. The DNA fork further cannot move, and hence replication may halt or completely stop. So this topoisomerase has a very important role for this activity. There are two types of topoisomerases, type 1 and type 2. Type 1 forms single-stranded breaks or nicks and then relieves this topological stress. Topoisomerase 2, or it is also called as DNA gyrase, it forms double-stranded breaks and then relieves the supercoils. So this topoisomerase enzyme plays a very important role either in DNA replication or in transcription. Wherever there is an activity in the DNA, the supercoiling should be relieved by this topoisomerase enzyme. So it plays a very important role in the cell. So in this class, we have seen that DNA replication is a very important process in a cell. There are several enzymes and proteins involved in DNA replication. DNA polymerase 3 is the principal replicating enzyme. DNA polymerase 1 and 2 are involved in repair. Primase synthesizes a short fragment of RNA. Helicase enzyme unwinds the DNA. Ligase enzyme joins the DNA fragments, and topoisomerase relieves the topological stress. So these are the things which we have seen in this class. Thank you.